All right, so the first topic on the agenda is a status report of whoever wants to share what they are doing, what their status is, probably some issues they went in, like a very quick and brief overview. Anybody who wants to share, if not, it's no problem. The second topic would be on discussion of purification protocols on protocols.io. I could find most probably one for BST polymerase and the one from MMLV from Alex Brown. So I just found this protocol. I don't know if it's for the BST polymerase actually. So these are the two protocols I wanted, or at least the one for the MMLV, I wanted to discuss it in more detail so we can go through it. Like, why is this step necessary? What should I expect? What should I basically do? What do I need to take a focus on and stuff like this? So really feel free to ask questions. I have a lot of by my own. And the next topic would be just like a resources and literature share. And for example, I have some handbooks which I found on Sigmar Aldrich's site I wanted to share with you guys because it's like a very, very good introduction into enzyme purification, as I think. And actually, Guy also posted some paper in the Reclone forum and the Slack channel of Yoga, which are pretty interesting. And if you have stumbled upon anything, just put it in the group, and probably somebody is interested in it. And last but not least, if we
is there a place for the group? Like, where can we put stuff? Do we put it on the Jokel, on the Reclone, on both?
You can always put it on the Reclone forum. I think it's the best because most of it's like the biggest overlap for everybody if you put it in the Reclone forum. And we can repost it in Google Slack if we want, but Reclone forum should be something where we post everything, like the minutes as well and everything. Yeah. So, and at last, I wanted to just do a quick discussion on IT infrastructure. So, how do you get along with handling the Google Docs for the protocols on open bioeconomy? Did you have some problems finding information? Which information, if you had some stuff like this? Just if we have the time, it's totally optional. So, yeah. Okay. Does anybody want to share something? I can also share a progress report of mine.
I think there's not too much to share from BioBlaze yet. That Isaac is now Dr. Larkin, so he has some time now. Spend.
Yes, indeed. One thing I don't think I recognize.
Isaac, I really can't hear you.
You're breaking up.
Badly.
Damn.
Is it your microphone?
Maybe close out some tabs if you got them open. That helped me yesterday.
And being next to the microphone helped too.
Yeah.
Yeah, true.
Is this any better?
Yeah, that's so much better. Okay, you had some remark?
I'd say, Sarah, I think you mentioned that there are some students on. It might be good.
Okay, you're breaking up again.
Just do like a one section.
You want to do introductions of people?
Yeah, I think that would be great.
Yeah. Okay. What do you think, Catherine?
Yeah, we can do that. I wanted to skip it a little because it's a little late, at least at my time. But yeah, I'm totally fine.
You only sleep.
No, it's okay. It's still 7 p.m., so it's not
Oh,
bad.
okay.
All right, then I just start the round. So my name is Catherine. I'm from Germany, and the labs I work in are in Darmstadt. It's like right in the middle there. I'm a master's student of biology and bioinformatics, and I started a project on enzyme optimization of BST polymerase for RT-LAMP testing. Yeah, so. And I met the other Yogel people like Isaac and Sarah. Rachel is also their guy, and everybody, Scott from there as well. Probably they can tell you something about it.
So I'll go next. I'm Guy Edelberg. I work out of the Center for Research and Interdisciplinarity at the CRE. And I'm finishing my PhD on also kind of LAMP-related stuff on democratizing DNA detection. And I'm also really interested in making these enzymes cheaply and easily.
And you're in Paris, yeah?
Yep.
Oh, okay. I don't know where people are from even sometimes on all these groups.
I'm mostly from Israel.
Mm.
Okay, maybe I can go next. So my name is Harry Akligo, and I'm based in Ghana. I'm part of the Open Bioeconomy Lab group, and also leading the Hive Bio Lab, which is one of the Open Bioeconomy labs in Ghana. I'm also currently a master's student studying how to locally produce some DNA polymerases, specifically called DNA polymerase and fusion, and using that to set delayed multiplex reaction for diagnosing 4 different sexually transmitted infections that are prevalent in Ghana. Yes, I have been working with Jenny and the other teams on the open enzyme collection. Thank you.
So maybe I'll go next. My name is Scott Pownall. I'm based in Vancouver, Canada. I run a community lab. It's the Open Science Network. And I've been connected, I guess, with Free Genes for a few years now, helping out with a variety of things. And also connected with the Open Bioeconomy Lab based out of Cambridge, Jenny Malloy's group. So I've been interested in creating open source tools for biology so that we can drive innovation in the space. And so that's been my interest. And I joined the Jugal platform earlier this year because of the Open COVID-19 initiative and started helping out there, and then helped to organize this subgroup to be able to produce low-cost enzymes. And so we got the funding and just starting to purchase the reagents and things like that to be able to get the ball rolling.
I'll go next. So I'm Isaac Larkin, freshly minted PhD from Northwestern. I live in Chicago. I've been a core developer on the Free Genes project for about 3 years now, and got involved with Open Bioeconomy Lab about a little while back as well, and very excited about democratizing the means of biotechnological production, and particularly the little molecular machines that are enzymes, and making it as cheap and easy as possible to manufacture and purify those.
Go ahead.
Oh, sorry. Hello, my name is Robson Tramontina. I am from Brazil, especially here in the state of São Paulo, Campinas. I'm a PhD student finishing my my degree, but also I have a startup linked to the university called Ecra Biotech, and we are trying to develop COVID-19 diagnosis tests here in Brazil. We're trying to produce these in a very popular price, especially to public institutions, in order to, trying to help solve those problems. And yeah, we all want to make this democratizing those type of enzymes and products for the bioeconomy. And also I brought here Viviane and Tabata, which are my partners in the business and university, so we can all share the updates later on.
Hello, I'm Ravi Kumar. I'm from India and pursuing my PhD working on lignocellulosic enzymes for biofuel production. Besides that, I'm also interested in open source projects where I have collaborated with a few electronic enthusiasts. Along with them, I'm working on some open source projects.
Ravi, we can't hear you at all. Yeah.
Am I audible?
Not really.
It's a little louder now. Can you say something?
Hello.
Hi.
It's very unclear.
Hello.
Yeah, I can hear you now.
Okay. I'm Ravi Kumar. I'm from India and pursuing my PhD in molecular biology. For my PhD, I'm working on lignocellulosic enzymes for biofuel production. In addition to that, I am also working with few electronics enthusiasts. Along with them, I am trying to develop some open source biotech instruments.
Again, Ravi, what are you trying to develop? I didn't catch that.
We are basically working on real-time PCR.
Real-time PCR.
Yes.
And you're trying to develop the hardware or some protocol?
Hardware and software also. We have people from electronics and computer science. Different people are working on different parts of RT-PCR.
Wow, that's super interesting.
Is the goal to make it open source?
Yes. Our goal is to make it open source because in India, basic problem is many students are not able to afford these high cost instruments. So basically, our purpose is to make all these instruments open source.
Mm-hmm.
Hey, I'm Sarah Ware with BioBlaze Community Biolab in the Chicago area. And we recently got a mini grant as well for working on this project, Open Enzymes project with Jogol. And so excited to get started. We're also working, we got a couple of mini grants from Jogol to work on SARS-CoV-2 detection. And so those are, I guess, kind of the two main projects right now for BioBlaze.
So hello everyone, I'm Natalia, I'm from Colombia. I met some of you in the Bio Summit. It was my first time there last year going there, and it opens my mind to this new area of open enzyme and bioeconomy, open bioeconomy lab. So I started to think how I can be like a part of this from Colombia. So I get some funds to buy some lab equipment. I'm professor in a university. And the idea is to start to work with these enzyme productions for many applications, mostly biotechnology. But for me, it's also interesting to teach or how elements for explain others. How it works, because we have some difficulties even in universities to access to these reagents. So it is nice also to meet you all again.
Should I go? Hi, I'm Rachel Aronoff. I'm from the Open Public Lab Aquarium in Switzerland. I've been working with Guy on the Corona Detective project, the Jogol method to detect the the SARS-CoV-2 virus. And I'm really interested in expressing these open enzymes for that, but also in general, because it's a long time since I made some T7 polymerase, and at Hackwarium, we don't even have a cold room or anything, and I wonder how people do things these days with all the, I remember just doing all this fractionation and a lot of stuff in the cold room. So I wonder if in this open public lab, it really is feasible to think about doing this, or if I need to get a new deli fridge or something for the lab. But anyway, I'm really excited to be here and meet you guys. And hopefully we'll get all the enzymes as open source as we can.
Nice. So, anybody else who wants to introduce themselves? If not, it's totally okay. I would just ask you to put your name and probably like your email address in the shared notes for if you want to connect afterwards. Okay, so I
No,
think
it's, I'm meeting. Sorry, Catherine.
no problem. Ingrid.
Okay, so yeah. So hi everyone, I'm Ingrid. I'm from Colombia. Right now I'm a student in a PhD in health science. Right now I'm the scientific director in a lab, and like everybody, I would like to have an open source and be part of this project and help to have an open source of these enzymes. I think it's everything. Thank you so much.
Does Claudia, do you want to say anything?
I'm still trying to figure out how to make this work on my computer, so I can hear
Well,
everybody.
I can hear you, so you could introduce yourself if you want.
Okay. Hi, I'm Claudia. All right, wait, everyone can hear me?
Yeah.
Yep, totally clear.
Okay. Well, I am in high school and I've been doing work at BioBlaze, and I'm mostly just interested to learn more stuff.
Nice. So you're at a good place for that, Claudia. That's super cool. Super brave. Welcome.
Are you working at SoundBio?
BioBlaze? Or, well, was.
Oh, okay.
Did we have
sorry,
yeah.
did you?
Yeah, BioBlaze.
Okay.
All right. Tabata Vivian Yakov, do you want to tell something? If not, just put your name in the shared folds. It's just Scott and Isaac who did that before. And I can see that now. So we can still connect each other a little bit easier. Yeah. Anyway, anybody who wants to share like a picture or a status report of wherever they are with their project, if they have like an active project? I can also show you like a crappy picture of my stuff. Just a second. I go for the share screen. So, can you see that?
Not yet.
Okay, so it should be actually on a shared screen. So, now it should be. Do you see it?
No.
No,
Okay.
just see the splash page, connect, create, collaborate.
Yeah.
Oh, okay. Just a second.
While you play with it, I'll say what I've gotten a bunch of plasmids from Free Genes and from Scott and transformed bacteria, and then I'm not sure exactly what to do. Kind of thing.
Yeah.
I mean, there's a bunch of different protocols, and there's a bunch of different ways, and there's a bunch of different stuff, and I'm not sure what to do.
Yeah, and I think that's kind of what this whole effort is about, is trying to figure out for the different enzymes, because each enzyme is going to be different in the way that we purify it. And I think most of the enzymes have a histidine tag on, which is a fairly expensive way to purify using the nickel matrix. So particularly from Isaac's work, we're trying to develop alternatives that hopefully will be lower cost, so different fusions on it. Yeah, over the next little while, I'll be working on trying to optimize the different protocols for some of these enzymes. It's just, yeah, when you have so many enzymes, where do you start?
Yeah. I would start even just with BST, right? Like that's kind of the most important one, even. And
yeah, for the LAMP assay, yeah.
And then I just posted on the Slack. I found some stuff that have to do with because I think most of these enzymes are actually heat stable. We should, I think, be using that to at least do some kind of denature, like most of the E. coli proteins, at least. And then I don't know.
Yeah, I actually opened up Slack yesterday. It was just a full-on day for me, so I didn't see this conversation. So
it was an hour ago because I was just going deep dive into.
Yep. So yeah, I only just after the Open Bioeconomy Lab meeting opened up Slack and saw your postings this morning. So yeah, it looks like some interesting papers. So I think it's the thermal stable enzymes that people have been using, the heat denaturation. I've got to read that first BMP report that you posted in Slack to see.
And then the other two papers, it's different ways. Both of them are different, cheap ways of purifying TAC.
TAC, yeah.
They basically first do some and then
just
some
a second. Can you probably repost the paper? Yeah, in the chat. That's awesome.
Yeah, so I just did the first one.
Mm-hmm. All right.
I don't know if you can.
Yeah, I'll do it.
I don't know if you can
One of them is
attach.
closed.
I don't know if you can attach PDFs.
Yeah, in the shared notes. I'll put them in the shared notes. Yeah.
Yes.
Catherine, you can say what you've been doing.
All right. So you should see the BST polymerase slide now, right?
Yeah. Yes.
Okay, so yeah, here. So I did my first run of purification. It's already 2 weeks ago, and I had some other organizational stuff to do. So this is my status quo. And here you can see the SDS gel. So this is the fraction or some lysate before induction, at the endpoint of induction, the inclusion body, the insoluble in water insoluble proteins, the
Did you go into full screen mode?
I did already, actually.
Oh, I see the
you can go into full screen mode on Big Blue Button as well.
Oh no, I meant for your PowerPoint, because I see the whole slide viewers on the left as well as the navigator on the right, so it makes your actual slide quite small.
Do you
hard for my eyes to read? Yeah.
Oh, okay.
Maybe you need to start presenting. I don't know how it works.
Yeah, I probably should upload it or I just do it that way. Just a second. Because it's actually better to upload it, but I was too. So that makes it bigger. Like this. Oh, I will hate me. Oh, yeah. Yeah, but now you can see it a little bit better, right?
Yeah.
All right. Okay. So yeah, this is my flow through of the column. So like the stuff that was not bound by the nickel column. And I first washed 5 times with 20 millimolar imidazole. Then I did a more a washing step with 80 millimolar, and I eluted with 300. And I just put it this year for asymmetry because I was staining them in one cup, so I didn't mix them up. Yeah, so basically I put little less of the lysate in here because it was hard to pipette, and you can see that the concentration don't match. And you can also see that a 300 millimolar of imidazole, which I used for elution, is still too low in concentration in terms of that you get everything off, because here this is the BST polymerase, and you also have some impurities. So I further did concentration and additional purification step with the emicon columns, like the spin columns. You put it in the centrifuge. But I just had a cutoff of 10 kilodalton, so probably this got a cutoff. But these impurities will stay. So, or it could be as well like polymerases. So like duplexes of it or something, because it also fits. For the Western blot, I couldn't check this fact because the antibody was super old and it didn't bind, I guess, but I will do it the next time. And I tried it on a test model that where I designed the primer with the Primer Explorer because I didn't have the real viral sequence before. And I don't know if the primer are bound badly or if the polymerase is not working. But I guess it's the primers, because when I put magnesium ions in it, and it's up to 8 millimolar. Yeah, I rechecked it in my notes yesterday. And 8 millimolar, I guess Rachel titrated it out that it's like the optimal concentration for BST polymerases in most cases. And I see some amplifications or some amplification of the signals, or higher signal than without and with the controls. So
Catherine, have you tried this with commercial BST polymerase?
no, the money will arrive at the university next week, and then I can buy them at NEB. But I'm still waiting for the money to be transferred to the public.
Yeah.
Yeah. Anyway, that's my status. So I will obviously benchmark them, and I got a new test model that worked at the Friends lab with the commercial BST. So I will try it again, and then I can see if I have a working BST polymerase. So this one is actually a gene. The gene I ordered it myself, and I had a super high transformation efficiency. So with the free genes plasmid, I actually just had like 3 colonies or something. And but I didn't check the concentration as well of the plasmid. But with mine, the plate was full of colonies, so it was easy to pick. Yeah, and what else? From 100 milliliters of culture, I got like 2.5 milligrams of BST, but I was really wasting it a lot during working, so this can be bigger. Yeah.
How do you know how much to put in a reaction?
I just did like 0.2 microliters, 0.5, and 1. And thought
after
like,
you
okay,
concentrated or before you concentrated?
After I concentrated it. And I figured,
I wouldn't even know how to guess, you know what I mean?
You mean because of the 5,000 plus reactions?
No, I would. right here, right? Like you don't have any real like lamp amplification. So like I don't. Maybe it's not enough. Maybe it's too much. Maybe it's.
Yeah, I
How
will.
do
I will check
you
it
have
with
to
the.
even?
Basically the thing is, I just wanted to start at some point, and I figured out how much I would need for with a commercial one, and I just like did a range like how I would use it for a normal PCR. And then I just did it because I wanted to test it. I have it and I wanted to test it.
Oh, I understand. Yeah, but because
And
like, but when I buy enzymes, they come in units. It doesn't.
yeah,
I have
yes.
no way of knowing how many milligrams that is or how many micrograms.
So the microgram, so the concentration of my concentrated of any enzyme is easy to access. I did the Bradford assay in a nanodrop, so this was easy. But I don't know if I have to. I have 2.5 milligrams of stuff that absorbs like a protein in my solution. That's basically what I can say. And for the process or for the unit definition, actually this will take some time because you need some assays. You need to define the unit by yourself, basically, because there is no definition of it for a standard. But I will look up how NAB defined it or any other commercial one, and probably with a DCTP retention time or something like that. Or I have to figure out an assay how I can actually measure how
Like a simple way to to benchmark we should try to figure out a simple way to for us to benchmark the BST, but also like all the polymerases maybe.
Yeah, I think that's also a topic and a breakout room in the Reclone Symposium.
Yeah, I think there might even be like an SOP on somewhere on the reclone stuff.
Ah, nice.
Yeah, I'm actually from Biobasic ordering a protein quantification kit. So, I think kind of have to measure the different parameters, and then it would be good to actually have a standardized test for a specific enzyme that everybody uses to be able to compare. So yeah, it's hard to figure out exactly how much enzyme to put in if you don't know what the specific activity is. And so you need to know what the amount of protein is and how pure it is, and then have some kind of assay to measure how well it works.
Yeah, this is true. So this is still work in progress. I just went in blind so I can measure something, it was just like a first try. And it was basically the first time I did enzyme purification in this lab, and I needed to ask every step of the way where I can find some stuff. So it was really a messy setup as well at first. You can also see that my gel broke, it's like, it was late in the evening. I was very angry of that, but it still worked somehow. So, yeah.
Yeah, so there are some questions like Rachel's asking dialysis.
There are some questions in the channel. I would just quickly answer them. So there was no dialysis step. I just just a second. Let me check my lab book. I did a lot of steps of Amicon concentration, and I constantly put some storage buffer in it. So there is less imidazole and more storage buffer in my basically storage buffer. Just a second. Where is it?
Does anybody know where I can get pre-made plates for gel electrophoresis? I've got the Life Technologies mini gel system, and I traditionally use pre-made gels, but they're damn expensive. So I'd like to switch to actually pouring my own gels.
Mhm.
And they do sell the pre-made plastic cassettes, but they're like $11 a piece for. I was thinking of actually maybe laser cutting some thin acrylic plastic to try to recreate those. I don't know if anybody's tried that. Yeah.
Yeah, I mean, you need to do some fräsen. I don't know the German word, basically. Just a second, I will check it. You know, like some cuttings on the edges for the gums that you can really like seal it in the chamber.
Yeah. A long time ago, I used to make
mill.
acrylamide gels for S35, P32 sequencing, the old school Sanger method. But it's, those were big gels, and leaks were a real pain.
Yeah, with the acrylamide gel, it always helps me to put some this magic tape, how's it called again? Parafilm.
Oh yeah,
on the downside. So, and I always like really put them toughly in the gum so it stays there and polymerizes well.
Yeah, and I've seen some protocols where they use like agarose. They just put that in the seam. So they're actually, they take like the Novex gels, and after they've used it, cracked it open, then they actually put it back together, and they use either epoxy resin was one method, and the other one was just agarose to seal it.
Okay, to seal it. Yeah, true. The trick with agarose, I heard that from one PhD somewhere, and I remember. Yeah, which is actually really nice because agarose polymerizes easily, and then you put on the toxic stuff. Yeah, that's a good advice. Yeah, I will think about it next time. I will pour some next week. Okay. So I just go through the questions. Ravi, specific activity. Yeah, so we need to develop an assay for that, but I think first I just want to get this work. So, but this is obviously a big point. Robson, have you ever considered treating the enzymes with DNase? Yeah. I did that, so I did thought about it, but I'm not sure if it makes sense if you have DNAs in your storage buffer and you want to have a DNA amplification. I mean, you will still get some amplification, but your pattern will get cut. But not sure if it's a good idea or not. I would take it out for discussion.
If I may talk a little bit now, because I'm very curious about this, it's related to the ratio between DNA contamination and DNase contamination, because perhaps it's a good choice when you purify your enzyme in the first steps, adding DNase, but in a very low concentration in order to get rid of some DNA. And then you perform a heat treatment for longer periods, then you're probably gonna not have DNase activity anymore because this enzyme is very thermolabile. So so I just wondering how you guys work with thermostable enzymes working with DNA at the same time. Sometimes you precipitate with ammonium sulfate or some another technique, and you can carry in some DNA inside your protein sample. So I'm just wondering.
You mean for the cross-contamination problem while doing a test, or why would you address
problems? Like if you're bringing in DNA like from the E. coli, it can then amplify. It can then serve as primers. If you're doing like PCR, especially if you're doing something with E. coli or even with the SARS and stuff, like it's a bit dangerous. And then in these papers that I posted in the notes, some of them they do a DNase and even RNase too, and then exactly after you denate, you you heat to denature them and things like that.
Okay.
Not everybody does it. Like, it depends. It depends how pure. If it depends how pure your enzyme is. Like with the his tags, supposedly you're getting. If we are doing like nickel and his tags and stuff, I think that should not be catching almost any DNA.
Can you post one of the papers you mentioned where they use a protocol?
Yeah, they're both they're in the shared notes.
Perfect. All right.
Because those are specifically for tag polymerase, so it's exactly the same.
Yeah, these papers, yeah, true.
So what happens to the shared notes after we're done with this meeting?
We'll export it into a PDF and put it on the reclone or something.
Oh, cool. Okay.
And on the Slack, yeah.
So actually, people may want to change the @ symbol in their email address to like what I did with mine, because if it goes public, then there are a lot of bots that will scan for email addresses, and then you get spam forever.
Oh, true. That's a good idea. I will do that for everybody, so don't worry.
Okay.
Yeah. So this is basically my state. So the in silico part, we are just starting it. We are getting the computational time now. We are modeling the BST polymerase. There are some fragments in a acceptable resolution, also with a lot of intermediates. So we are pretty positive that we can probably have some like good results in January, something like this. And until then, I'm just developing the implementing the test models, implementing assays and stuff like this, doing networking. Yeah. So anybody else who wants to talk about their project a little, like show like a slide or just like 5 minutes what they're basically doing?
I don't have anything to really share at this stage. I've got reagents coming in, and so once those are in, then I should be able to start producing some tests with it. So yeah, I still have to get the nickel.
Do you have a list of what you bought?
It's pretty much. Yeah, it's in that joggle, but because I didn't get the full amount, I'm revising it a little bit, and I'm rather than using pre-made SDS page shells, I'm exploring, pouring my own. So trying to figure out how I can make the plates without paying the ridiculous amounts. Somebody's given me a couple of the blank cassettes for. That are the same ones that the Novex gels use. They sell them in the Canadian Thermal Fisher for about $11 a piece. You have to buy 25 of them, so quite expensive. So I'm looking to see if I can actually design, laser cut the plastic. I just don't know. If laser cut acrylic has the same thermal characteristics as the injection molded stuff that they have, but I'll give it a go, see what it's like.
All right,
I think something that might be good for us to do is make a document, maybe a Google Doc that tracks all the stuff that we've ordered and if it's arrived, and that way we know what each other have, and
okay,
if one person is short
Isaac,
stuff,
that's a good idea, but let's discuss it in a Yogel meeting.
Okay.
Yeah.
Sorry.
I actually started tracking that, so I do have that. So it's just a matter of sharing that. But I'll wait until more things come in.
It would be useful, even for me, because there's so many different, even like nickel columns and resins and sizes and things, and I just don't know where to start.
Yeah, but I'm actually, because I have some blank columns, reusable chromatography columns, I'm just getting the slurry from Thermal Fisher. It's 10 mils for like 180 Canadian. That's what I could budget for. but you can actually reuse them. So, you know, it's not a one shot, and use 2 to 3 mils of the slurry for one purification. And then there are also protocols for how you can recharge the nickel, so that you can extend the life of the pellets. I haven't tried them, but Alex Brown has actually been helpful with that.
In my lab, we also have some enzyme purification people, and they also help me a lot. And one of them is doing it a lot, like basically after every run, because they always just need one purification run of an enzyme and then, or a protein, and then they do another. And after a run, they have a very good protocol. I will just ask him next week, and then I can upload it in the Reclone channel, or in the Reclone forum and Slack channel as well. I will just put it as a to-do for me. And yeah, so if anybody's interested, so it's basically like a lot of the nickel resins are rechargeable, and you can use nickel cations, but also copper and cobalt and different other ones. But basically, it's a good point to start with nickel. But still, you always have some residues in your resin with your old enzyme or anything. So you need to take care that everything goes flushed out, and you put a liquid with the nickel again, and the nickel binds to the binding sites in the resin again. And yeah, then you can reuse them, and they should have just a little impurities, but it should be enough for our purposes. At least for research and cutting costs a little in there. If you will produce them, you will basically use the same column for the same enzyme all the time, so this doesn't count too much in there. But it's still a good idea to strip them and recover them for after some time, if it's possible. You have to check with the manual actually of your supplier. So, anybody else for some updates? If yes,
I just now have an update.
Isaac has one.
Yes, I revised the budget for the BioBlaze contribution so that reflected, instead of having a $3,000 budget, $2,000 budget. And I just
Isaac,
sent that.
Yeah. It's not a yoga meeting. We are with the Reclone people, and it's basically just enzyme purification, you know?
Okay,
sorry. But it's, yeah, we can discuss it soon.
I was confused about that too, but yeah,
Okay,
thanks
sorry.
for clarifying, Catherine.
Okay, so Robson, you have also a COVID-19 test project, which I can see. So you're also developing a test. Is it also RT-LAMP based?
We're actually trying to develop both at the same time, the RT-qPCR and the LAMP. We did not yet started producing the BLST. We are just expressing this, and we have the pellet. So we hopefully we're gonna try a purification next week. That's why I'm very excited to have this meeting with you guys. We just developed our first, let's say, two-step COVID-19 test for RT-qPCR, for trying to validate our enzymes separately. And our next phase is going to be building this one step. So I have a lot of doubts how to design the best experiment. So I would be very grateful if someone
Are you
is
in
also
contact with the people in Chile and Argentina from the Reclone? Because they are already kind of succeeded, supposedly, to do this.
I'm talking with a Mexican guy who has a great transcriptase, reverse transcriptase, but it's nice to know that they are having success with those open source enzymes. We are very excited to share our results and get some help as well, because it's very tricky when you're wasting a lot of time. And you know, as everybody knows, Brazil is suffering a lot with the pandemic, so we are rushing to develop this as soon as possible.
There already is one that supposedly works well for the codon optimized MMLV and the mashup reverse transcript.
Oh yes,
up on the.
We have an enzyme that works very similarly to mashup. It's very head stable. It works well at 60 degrees. We got a guy from the Pipette Jockey. It's a website.
Yeah, that's the mashup.
Yeah, the mashup. Yeah. It's pretty good enzyme. We validate with COVID human samples, and it works pretty well. So the next step is going to be trying to develop a hot start method for TAC DNA polymerase, so we can mix it up everybody and see what happens.
Like you want to make your own sort of aptamer for the TAC?
We have an aptamer here, but we will try as well those called SSB protein, single strand binding proteins. Hopefully we can see some comparison between of them, but we are betting the examers are going to work better, but we didn't have tried yet.
Sounds
cool.
Yeah.
There is some patent implication for aptamers in use with BST. I know that, but I mean, it obviously depends on the aptamer and on the region. So I just wanted to.
And the country.
Yeah.
Most things are not patented everywhere.
Yeah, that's true.
Here in Brazil, we mostly don't have any patent protection from any thermoscientific. So it's basically we can operate. Even RTX we can express here and produce without having any complications.
I know the mashup, the guy that made the mashup, he doesn't want anybody to make any money using his enzyme.
Okay. Oh yeah, I'm in touch with him. I'll let him know about the situation we work with mashup. But we do have other option as well. Superscript 3 here is we can produce this as well. And superscript 2.
Yeah.
So superscript 3.
The 3 will be, but it's not yet off patent.
Yeah, superscript 3 goes off patent in January next year. So it's pretty close.
But again, it depends. In each country, it's different. So it really depends.
Oh, yeah, that's also true. Yeah, I'm speaking for Europe now. Okay.
From America, probably even yeah. I don't know.
Robson, how do you check for patents actually?
Actually, I checked. There's a website. Actually, my coworker do this for me. He puts this DNA sequence and see what happens. But also we just search from Google Patents and the national patent system here, INPI. But sometimes when you are in very doubt about how it's gonna work, we have some extra help for PI specialty people. So they came with a sort of report for us and say this is okay to work with, this is not okay, mostly.
That's good. So always take care that you are safe.
Yes, we are very concerned about anything about this.
Nice. Yeah. Did you guys attend, or you, Robson, as well, the last Reclone Symposium, actually?
No,
so
I was unaware.
No problem. So it's there was a talk about patents as well, and they also had some resources mentioned there. And you can find all the talks on YouTube, actually. So I will post the link in the channel, or Harry wants to say something.
Yeah, I was going to say that one tool you could use for patent search is the lens.org. It's been linked to quite a number of the patent offices, like the US patent office, the Japanese patent. So you could use that to search in quite a wide range of databases that document most of these patent documents. And
it's
I
good,
mean,
and
if
it has
you
the
are,
families and the histories, and it's all kind of
yeah,
connected.
they have quite a lot of tabs that will help you to streamline and search all those documents and to know that the enzymes you're working with are off patent. But I think the Open Bioeconomy Lab team has done quite an amazing work on that front. So and we have most of the documentations on the Open Bioeconomy Lab website. So if just in case you are looking to know which enzymes are off patent, you obviously could check out that just so you can save some time having to search most of those things all by yourself before going to. And obviously you can ping in some notes some notes in the Reclone forum, and whenever people would just come up and tell you whether these enzymes are off patent or not. But if you want to take the time to search, I think lens.org it's one of the best platforms to use. I have also used that quite a lot during the start of the project, so it's an amazing tool to use.
Yeah, thanks, Harry. I checked it out once. I actually found it much more structured than Google Patents, and it has also like this it's a different setup or presentation of data. It's basically much nicer than just the raw data in Google Patents. Okay, so I would suggest we go on to the next topic, or does anybody want to add something somewhere?
Hi, I'm Natalia. So far, I've collaborated with a professor. We obtained, thanks to the Open Bioeconomy Lab, the RTX retrotranscriptase from Allington's lab, and we can obtain it, and it is working. The one that is exo minus, if I'm not wrong. It's working really well with the diagnostics part.
Does anybody know if that's the same RTX that like NEB sells, or they're different things, right?
It's so far what I know that it's a synthetic one that they develop in the lab. So it's the new one. They have two versions of it, like Exo Plus and Exo Minus. They send you both if you ask for it. But the one that works for us is the Exo Minus for the COVID diagnostics.
Yeah.
So the
PCR,
BST.
you're doing PCR.
Natalia, are you doing PCR? Or
we are doing qPCR.
Okay. All right. Cool. So yeah, basically, and you now want to try to purify the enzymes yourself as well, because you have problems assessing reagents for the PCR.
Yeah, it starts with the idea that, I mean, I got interested in these topics the last year, because our difficulties generally to access to reagents, but of course COVID situations put us in a rush to do it. So we are producing it. The idea is to help with this, but of course we need validations and approvals to do it in with patients and move forward. But so far the research that my colleague has been done with the plasmid that I can get being part of this network show positive results that the efficiency of the enzyme, it's comparable with a commercial one. I think that someone is asking that if BL21 is the strain. Yes, I guess that it depends of the plasmid, but for these enzymes that we are using BL21 strain.
Like a special strain of BL21, like the Pylus or the Pyril with a special codons or Rosetta, or just the plain BL21 DE3?
So far, the plain BL21.
Yeah, the other ones are very expensive as well. So, yeah. Okay. So, yeah, the Rosetta strain is also part of the purification protocol from Alex Brown, which is also the suggested purification protocol from Free Genes and Open Bioeconomy. I just put in the link in the public chat, so probably you all.
I put in another one that I know people are also using there, like under for the MMOV and the mashup, which is codon optimized and uses the not the Rosetta.
So I would start with this protocol, and probably we can go through the other one and compare to discuss a little. Okay. So I just post in the link. Yeah, and Harry just reminded us on the Reclone Symposium on 29th of July as well. So it's on the page. You can register yourself already. Okay, Ravi. So nice to meet you. Have a good time. Bye. So if you all could open up the protocol on Protocol.io, I posted the link, and we just go quickly through with it. I just give you another minute to open it up. Ingrid also needs to go. All right. So, now let me just check. Oops. Yeah, I would share my screen again. No.
It's been updated to version 2. Do you have any idea what changed?
No, I didn't do a diff, or I didn't compare. I just have this one issue of when I want to share my screen, I don't know why, but this time or probably I open it in another browser, because I can only share my tab, which is the actual one, and then everything goes to hell because I share the screen of the big blue button screen, which is not good. But just a second. Yeah, there it is. Okay, I just share the screen of another folder. All right, so it's for the MMLV, and it's like the recommended protocol for purification. Yeah, let's.
You're doing the same for BST, basically. That's what you're saying, or?
Yeah, there's not a big difference. I actually used this protocol that was provided for MMLV for BST purification. It's both his-tagged, yeah, but I don't go into purification optimization too much because I just optimize the enzymes. But anyway, let's just go quickly through it. So you do a transformation of your plasmid on, here it is the Rosetta strain. You use Rosetta when you have codons that are rarely used. So for example, the MMLV, it's a protein that originally came from mice. So it's from a mouse of mouse origin, and they sometimes use codon. Bacteria doesn't use so often, so you use this Rosetta, which is basically BL21 with a special plasmid that codes for for other tRNAs. So you can just have a bigger yield actually. But as Guy said, if you have a codon optimized version and it's functioning, you don't need that anymore, which is nice because they're expensive. So you do your overnight culture in 5 milliliter of LB broth, for example, and put your antibiotics in it. You, in this case, you use two, one for the plasmid with the special tRNAs and one for your plasmid with the enzyme on it. And then, yeah, they do a 500 milliliter LB expression. So basically, that's one thing I've learned from our postdoc. They usually do 100 milliliters in 1 liter flasks because the inoculation, so like the mixture of oxygen in the media, is like the best in this ratio. So if you have just 1 liter flask, it's better to probably just put under 500 milliliters in it. It's because they really need the oxygen to grow better, and then if they grow better, you have more yield. So that's easy, and obviously you sterilize them in the autoclave before. If it's you can just prepare it easily. And then with the induction, so the induction step is like the first step where you get probably overwhelmed by the possibilities you have. So I'm like a more pragmatic person, and I always screen for the minima and the maxima and go somewhere in the middle. Basically, it doesn't depend where you start, but you have just put it in your notes and compare if you have a bigger yield or if your protein is not working well, and then try a different parameter until you're set. There are better approaches of doing that, but this is not part of the discussion today, I would say. So Guy actually suggested it to me the first time I met him. It's called Design of Experiments, which is a super method. I read into it. I'm totally a fan. But yeah, you need a lot of time and a lot of machines or hardware for it. So, okay, so you take some of your overnight culture and you put it in your prepared flask with a big volume of media. And then you start to shake it without the IPTG, which starts the induction of the expression actually, until it has a OD of 0.8 to 1.0. So 0.8 actually is perfect. Some protocols say just take 1 milliliter. Some protocols say dilute the overnight culture to an OD of 0.06 or something. So basically, I would say this is like up to
everything I've seen says like do 1 to 200.
1 to 200?
Like to dilute the overnight culture.
Okay, so you just, for example, if I have like 200 milliliters, I would take 1 milliliter, and yeah, that's also a good point to start. So yeah, what is important, incubate it at 37, otherwise you will wait until you are gray. And what you also have to keep in mind that you don't have any antibiotics in your bigger culture now. So try to work pretty sterile if you take if you go in with
Why you do? But you in the big culture you do have antibiotics.
in the big culture.
Yeah, it says supplemented with carb and cam. You can put the antibiotics in the big one.
No, here.
3.1. No.
Here. Oh yeah, okay. So we don't put antibiotics in the big culture actually, because we just take care that we work sterilized. And I didn't have any problems with that actually. So yeah, basically the problem could be that some of the bacteria lose their plasmid because they don't have any pressure.
That's why I would put the
to keep it. Yeah, but I don't know. It's just the protocol. I mixed in the protocol from my lab a little, but that's actually a good discussion, right? So yeah, probably I do it next time. Actually, it doesn't matter so much that I didn't put it in. Anyway, so take care that you take a sample of your culture right at the point before you give IPTG to it, and take a sample of your culture right to the point after you take it out of after the long period of expression. And yeah, so basically that's it. The monitoring of the OD, it's actually a step that comes in here. So you do it before induction and after induction. They say 0.5 millimolar IPTG. The range is huge, is from 1 molar to 0.05, and it's really depending on your system. So 0.5 is a reasonable way to start. And after that, you go down to 80 degrees and induce for a lot of hours, which is basically better because your proteins are folding most probably better at a lower temperature. So that's why they go down. And obviously the bacteria is not growing fast and producing fast. So your expression time needs to be longer. But 20 hours...
So also I have a question here because I've seen lots of different protocols. Like some people say 1 hour, 3 hour, at 30, at 37, at 18, for 18 hours, for 24 hours. Like I've seen it all over the place.
Yeah, so I mean, all the way will work somehow. It always depends how much time you have, how your workflow is, probably. Yeah, anyway, but if you express at 37 degrees, then the protein folding could be, so you could have too much kinetic energy in your system, so the protein folding not most of the time, but more often than with a lesser kinetic energy at 18 degrees Celsius.
Even with the thermostable protein? I don't know.
Yeah, so it's about the folding process, not about the thermal stability.
Yeah. Okay.
It's about
how slow
when you fold it more slowly, it's easier, that it goes in the right direction. When the folding is when the folding is lower or at a lower temperature, the probability that the folding succeeds is much higher most of the time, not always. But it's reasonable to do that at 18 degrees Celsius if you have the time. But it will also work for 37. Just make sure if you change something in your parameters and you benchmark your protein or you do the specific activity assay or something, that you do it again. So you have consistent results, because it could be that you have less active protein after when you express at 37 degrees. So
less protein or more protein, or we don't know.
Yeah. So the specific activity would vary, so you need to do the assay again. So yeah, that's always something to keep in mind, that you have like a population of proteins in your solution, right? Some of them are badly folded, and some of them are folded that they work a little. A lot of them would be folded that they work super efficiently. And we cut off this recycling process of the cell. So we stay with this population we have at the expression endpoint. And yeah, if you have less kinetic energy, it works usually better. Okay. Any questions so far? So Guy's actually doing like a good job when he's just like throwing questions onto me, and yeah. Okay, Ingrid has actually a question. Is she still there? Yeah, she's still there. She said she had worked with Pitchia Pink. Okay, what is Pitchia Pink, Ingrid? Could you explain it to me, probably? Or she's typing, she didn't has... Pitchia pastoris? I just Google it. Just one...
Actually,
it's a fungi.
yeah, it's an expression. Yeah. Oh, sorry. It's an expression vector. So I use it like to process one protein that I was working with in my PhD, and then I have good results because it produces like a kind of back wall. And it's easy to purify then the protein, right? So what I was asking is that if you have expertise in this area, if you have worked with a pichia or another kind of eukaryotic cell, and if it's better to work with a enzyme expression and everything.
Okay. So I have never expressed proteins in yeast cells, actually, but I have a colleague who do so, and I just know when you need like the secondary modifications, like glycalization and stuff like this. You use the easiest eukaryotic model system, which is one of the yeast strains, to do so. So, do you purify in yeast and your proteins for your COVID-19 test, or do you just do it on a regular basis?
No, it wasn't a protein to. I don't know if you know this protein, but it's a APO. The name is APOL. It's a normal protein in the human blood, but I'm working with that as a treatment. So I'm producing that for the treatment in animals. So that's why I was working with a pink. And but the problem that I have with that is that everything is attached to Thermo Fisher, so I have to produce everything with all the products that they sell, right? So it was kind of difficult, the situation, because everything was so expensive.
Yeah, I can totally understand that. So for the enzymes you need in biolabs, you don't need the glycalization in general.
These are all bacterial enzymes. So
Mhm.
we usually
Yeah.
express them in bacteria.
Yeah. So there's, in my opinion at least, but I'm no expert in this, there's no need to use yeast as expression system. But for your use case with the APO protein, for example, it totally makes sense because you need the after expressing processing of the protein, you know, the glycosylation and the Golgi apparatus and everything. And this is not happening in bacteria. Which is good for our purposes because the BST polymerase, for example, is a bacterial enzyme, so it doesn't need that. The MMLV is a eukaryotic enzyme, so I don't know if it's worth a try to express it in yeast and see if it's more stable or if it's somehow improved. I can't say that. So anybody some opinion on it, probably?
Thank you, Catherine.
No problem, Ingrid.
Just
But,
on the MMLV, it seems that a lot of people have got it to work in bacteria. That's all I can say.
Yeah. So, and it's the easier system, that's true. But Ingrid, I think you also put your email address, and I can, if you have some questions on yeast purification and how to use a different model and everything, I can set you up. I can ask my colleague who's working with it if they have some advice for you. I don't know if they use also the proprietary reagents from Thermo Fisher, but probably he has some advice that can ease the cost a little. So I'm just checking. Yeah, I see there is your email address. I will ask him on Monday, and then I will just write an email to you if he's okay with it or not, because he's not connected to Reclone or anything. So I'm not sure if he has the time, but if he has, you have somebody to ask some questions.
Okay, Catherine, thank you. I would like to say something. I met this reclone community by Natalia, and Natalia showed me how it works and how you do everything. Actually, I'm working in a lab, and we were working to try to purify and to make tag polymerase because it's really difficult in Colombia to have the reagents here, and sometimes it get lost and in the shipping. Sometimes arrives really in bad condition. So that's why we would like to have a kind of project and have something like the thing that you want to do, that is an open source enzyme purification, enzyme network to try to work with and to try to diagnose, because actually I work in a diagnosis lab. So we would like to have something like that and to make research too. So thank you so much for that.
Yeah, no problem. It's the Open Bioeconomy Lab who started it, and the people from Yogel. So yeah, but do you have problems setting up the purification, or is it just like your first step that you just thought about? Okay, we can do that as well, and we want to hop on now.
What I tried to do, it was to design. I make with a company that the name is GenScript in United States. I tried to design the gene inside a vector, and they already give it to me. And I tried to use an E. coli BL21 to produce it. Is with the Thermo Fisher 2 company, but actually it didn't work. I mean, I don't know. We have to make a lot of tests. Is the first step So that's why I'm here to try to improve the protocol and to try to help us to do it.
You can also send us the sequence data, and we can check for that if you want to. So, where was the problem? Did your bacteria not get transformed with the plasmid, or didn't you have any activity, or where was the bottleneck right now?
I have no activity.
Okay.
No activity. Yeah, it was a
Yeah.
problem because actually I think the bacteria get with the vector because it's the blue color and everything. So I think is transfected. It's just the activity that is not doing it. Is not working. So.
And how did you check for the activity? You wanted to purify Taq polymerase, right?
Yeah, that's the first step. I mean, I'm a baby in this kind of area, so
We all
I'm trying to do it, and that's my first step.
Okay, so don't be disencouraged because of that. Like, I've heard that for everybody, the first run goes wrong. You know, my run probably also went wrong. You just have to hop on, reflect on your mistakes, probably talk to people like we do now, you know, but it will work someday. It takes some time, obviously.
Okay, thank you.
So don't be disencouraged. It's a lot of mistakes you need to do until it works.
Okay.
I want to add something that like Scott posted before in the chat. It's a link to the Free Genes collection.
Yeah.
They will send you free enzymes, like free plasmids that have the open TAC and MMLV and BST, and like it's a bunch of different already plasmids with the enzymes on them. If you want to try that, they send it for free anywhere.
And it's 42 different plasmids, so just go on the link.
There's that's the open. There's a COVID diagnostics toolkit enzymes. Those are already on expression vectors, and then there's also the open enzyme collection is on different plasmids, but they can send you that too.
Yeah, I also got a lot of plasmids from them, so this is a super resource. It's like a core part of reclone, I would say.
Yeah,
I think.
Yeah,
I'm not
that's
part of the reclone people, so it's just how I see it from the outside.
That's the core of it. That's what. Yeah.
All right, so for everybody, if you need anything, order it there, and we can also try to share some plasmids somewhere, or just send them to Free Genes and they share it to everybody, which is nice to have a central hub like this. But really surf the page, so it really helps. Yeah. Okay, so we were at the point 4 of the protocol. Or does anybody want to add something? Or does anybody have some questions so far? Or probably too much? Okay, but I will just continue because time runs a little. Okay, so we are here. Now we have our flask of 500 milliliters in this case of expressed enzyme in E. coli cells inside. And now we need the enzyme to come out. So first you collect the cells, you put them in falcons and centrifuge them. So you have a pellet with your bacteria. And this is the collecting. Then you harvest the cells. So you actually you have your pellet, you put away the media, which is not necessary anymore. And then you want to crack open the cells, right? So the protein gets out in solution. And this is accomplished by washing with lysis buffer. So you put some imidazole buffer in it. And then it is important to put, or I don't know if it's so important, but in every protocol they say it should be ice cold. You put the lysis buffer again, like 5 milliliters per gram of wet pellet. I think I did a little less. No, I did 5 as well. And the lysis buffer is, it's basically the equilibrium buffer for the column, which we will discuss in a few. But you add PBSF or AEBSF to it, but you have to do it freshly. Otherwise, the reagents get oxidated or reduced and are not active anymore. And this reagent you put in for proteases inhibition, because inside of E. coli, you not just have your expressed protein in a higher concentration if you're lucky. You also have different other proteins and enzymes, and one of them cuts proteins, or a few of them. And they get inhibited by AEBSF, which is the safer but a little bit more expensive alternative to PMSF, sorry. And so you got this problem covered, and then you put lysozyme in it as well.
Where does it say this PBSF stuff, or?
It's
in a different one. I saw PMSF.
Yeah, it's basically it's the same concentration, but AEBSF is less toxic than PMSF.
Okay,
So,
they don't in this protocol. They don't say that they put it.
it's the lysis buffer, and I think when we get to materials, for lysis buffer, you add it here. That's
okay.
So these are the buffers. They basically, so these buffers differ in the imidazole concentration, which is important for the elution of the protein. And
water with imidazole, lysozyme, and
no, you take the buffer B. You take the buffer B here.
Yeah.
This one, and you put freshly REBSF and lysozyme in it, but you have to do it on the same day. And
that's not written well. Okay, I would never be able to understand that. Okay,
no problem. So that's why we are discussing it. Yeah. So you take this buffer, which you also use for your column later on and for washing a little and stuff. And you put lysis buffer in it, and you weight your pellet from the centrifugation step here. And per gram of pellet, you put 5 milliliters of the lysis buffer on it, and then you have to resuspend it, or you already resuspended the pellet in lysis buffer, so it's easier for the to distribute it. But at this point, it gets like really sticky. So it's better to put it on ice. That keeps it a little bit more fluid, actually. But
I found it a little hard to resuspend it.
Yeah, it's not nice. So what helps a little is when you have your pipette and you cut your tip a little. So, for example, when you pipette Tween 20 or Triton X100, you also take your pipette and you cut the tip, but not straight, but a little in an angle. You know what I mean?
No, but usually for this, I'm using like big pipettes.
Okay. I used to.
I used like a big like 5 ml or 10 ml pipette because it's bigger volume.
So you mean the glass ones?
The hard plastic ones, the big
or
ones.
hard plastic ones. Yeah, I could try that. I always used like 1 ml or 5 ml of the normal pipettes with the plastic tips in the boxes. I probably try that next time. Probably it helps me a little. Yeah, so with the lysis, you crack up the walls, or it helps up to crack up the walls of your bacteria, and the AEBS, the lysozyme, and the AEBSF inhibits the protein cutting enzymes inside there. So you are set to, somehow helping so it's not like the lysozyme also helps to crack up the walls, but you need some mechanical stuff happening in there as well. So the walls just don't get cracked up by the lysozyme by itself. You also need, so there are basically different methods. You can do thaw and thaw it in the cold and put it warm again. But I've read at the Free Genes information paper that this thaw refreeze method is not recommended for BST polymerase, at least because it sets down the activity. So I don't know if Ingrid, for example, if you have like so how do you call it in English? Actually,
I don't have a sonicator either. Yeah.
How do you do it then?
I don't know how to do it.
Okay, so you can try it with the freeze.
I have some reagent that says like that. That's what it does. Like it pops open cells. Like also some kind of thermal fissure, like liquid.
Okay. So but it would be lysis would be lysis is also a thing in RT-LAMP protocol development, but lysis for enzyme purification also should be a thing we should probably investigate a little more. So I did it with sonication. So you have like a huge sonifier which does like sonic waves on the tip, and you put it on for 10 seconds, you put it off for 30 seconds, and really need to cool your sample because it really gets warm quickly. And you do that 15 times, which is, nice. But you can also just like freeze and and thaw the sample as well. Ingrid, how did you do it actually with your purification attempt?
I'm using a kit of thermal fissure because I have not sonification method. I mean, I have not sonification protocol and equipment.
Are you using this PBER, complete bacterial protein extraction reagent?
Yeah.
Yeah?
Yeah.
Try that.
So probably this is also something we could think of, that we take this protocol and we see what other methods are open which don't need this heavy hardware like a sonifier. So I just put it in on my to-do list. Can you say the name of the kit again?
It's PBER. No, BPER. Protein extraction. Bacterial protein extraction reagent.
Bacterial protein extraction reagent. Okay.
I can give you like the reference is 89822 from Thermo.
89822. Okay, all right. So probably we can also find something there. But yeah, let's see how fast we get on it. Just a second. Yeah, all right. So yeah, I did the sonification protocol because I was lucky to have a sonifier at the university, but this is a huge point. We need to take care of this. Yeah, then you spin down again, so you have your soluble proteins in your liquid, and in the pellet you have the inclusion body or the insoluble proteins. And both of these are the next two samples you take. So you take, for your SDS gel I showed in the beginning, you take one before induction, at the endpoint of induction, one of your soluble proteins, and one of your inclusion bodies. Yeah. So you also need that to compare everything, your efficiency and stuff, and try to really, when you do it, the protocol the first time, it's better to take more sample than less. So if you take like 80 microliters, you will still have enough for purification. But you will be angry otherwise if somewhere some sample is missing. So if you are like adjusted to the protocol, you can also go calculate like for efficiency. But in the first steps, I would recommend you to always have a mistake margin in the volumina you're sampling. Okay, so yeah, which is always better? Or is there any question until now? Okay. So, yeah, it's always better for the elution step now. Now we are at the purification. So this all was just like producing the enzyme and getting in touch with it. And now we want to like really purify it. And IMAC is like the immobilized affinity chromatography. So it's basically like you have your nickel cations that are immobilized in the resin, and they are very affin to the his tag on your enzyme. And if you put imidazole in it, it breaks this interaction between these two. So this specific interaction goes also flows through them. And that's what you try to exploit in this method, actually. So you take your lysate, but you take your clarified lysate, and that's one hint I got in the beginning of enzyme purification. It's always better for your column to filter your stuff. When you filter it, all the particles which could like stay in your column forever and make your column unusable somewhere soon are not in your flow through or in your lysate you give onto the column. Yeah, a column basically is just like a pipe, and down on your pipe you have like a little hole, and up the hole you have the resin where everything stacks, and you just pour it inside basically. And when you do the columns by your own, it's also a nice thing to put a second filter above the resin. So when you pour your samples or your buffers inside the column, you don't mix the resin again, because otherwise when you mix it up again, you need to wait until it's set, and it's also not good for the material if you mix it around too often. Yeah, so yeah, how to take care of the column? You basically store it in 20% ethanol, but it has to be like the pure ethanol and 80% water. And then you can, if you take it out of the fridge where you should store it, like sealed, so it doesn't lose too much of liquid, you first put some rounds of distilled water in it, and then you equilibrate it with the 20 millimolar imidazole. And you don't use the lysis buffer. This is actually a mistake. You don't need the lysozyme and the AEBSF in the equilibration part. Now you just use this one, only these ones. So this is important. Yeah, and then you can, if you did the equilibration of the column, you can put your lysate in it. It's better to filter it always.
So can you say one more word about like equilibration, like to equilibrate it? It means just to leave it together for like swimming in it for a while, or?
No, it basically depends on your column volume. So let me just check how much I needed. I think I used like 5 times the column volume for equilibration. So I have in my column, I have 1.5 milliliters of resin. I did it myself and I put 1.5 milliliters on it. So my column volume is 1.5 milliliters. And basically how much buffers you use always depends on your column volume. So you always have 5 times, 7.5 times your CV column volume. And yeah, here you can see it, 10 column volumes of buffer C, for example. And there's also, like from Sigma Aldrich, there are also handbooks on enzyme purification, and they are really helpful. I will post the link soon in the chat.
I posted two of them in the shared notes.
From GE Healthcare.
Yeah, there's the regular. There's like the
the handbook.
And then there's also the recombinant protein purification.
And I really can recommend these two. They're super. So they give you, of course, they want to sell you their products as well, but they also give some very good advice. So it's always nice to follow these manuals.
But just again for the equilibrating, it just means like pass through like this amount.
Yeah, it's just you pass through like up to 10 column volumes of the buffer B after you washed it with 10 column volumes of distilled water.
Okay.
That's it. Then it's equilibrated.
Okay.
You don't need more. So it's also about the salt concentration and everything. Yeah, just a sec. Here, I'm lost in Windows. Yeah, and that's it. You just put it in. You can basically, it depends how long your pipe of your column is. You can basically, like, I don't know, let's say I have 1 milliliter column volume and I want to put 10 CVs in my column. I can just take 10 milliliters, put it in there, wait until it's finished. It's super easy. It always seems so weird, but it's basically super easy. Just make sure that you don't disturb the resin. That's basically it. And that everything is filtered. So what makes actually sense when you set up your experiment is to make a 10 times concentrated version of your buffer, but only with the these
same, yeah. Except the imidazole is different, yeah.
Only with the tris and with the NaCl and the triton. But you do 200 millimolar, 3 molar, and 5%. And then you can dilute it with imidazole as much as you need freshly, because imidazole is also reacting in the. I mean, it should be stable in the fridge for 2 years, but it's always better to keep it not with salts and with a detergent. So you keep it in just pure imidazole, and then you can add it freshly. when you dilute it from the 10-fold concentrated stock. Yeah, so this is actually always something you should consider. And then you put one part of water and the other part of imidazole and the rest water, one part of glycerol. And if you don't have glycerol or it is too expensive for you, it's also okay. I think you put it in there so the viscosity is a little bit higher, so it doesn't flow through too quickly. And there's also some hydrophobic interactions that can help the purification process, but I didn't get into detail with that. So that's optional. You can put it in there or you can't. The detergent should be in there. It's just easier if you set down the tension of the water surface. I actually used 500 millimolar of sodium chloride. Still?
Yeah. Now you're back.
Yeah, I got kicked out of my internet network. Oh no, and now everybody is going. Okay. So, yeah, for the people who are still there, I would continue. All right, just a second. Yeah, so you wash it with the 80 millimolar, that's also optional, and then you elute it. 500 millimolar imidazole is like the highest concentration you should use. I used 300 and I still had some BST on my column. So next step, I would go higher. Again?
We don't see your screen anymore. We don't see the protocol.
Oh, oops, sorry. True. Yeah, but it's also long. Here you go. All right. Yeah, and that's basically it. You collect it in little epis, and then you just change the epi. When it's down, after that I used. I was measuring the enzyme concentration in a Bradford assay. Also loaded my samples from every fraction on an SDS gel. And cooked them before, of course, in Lemley buffer and run the gel. And also did a Western blot that didn't work out. So I know the sizes of the protein and somehow-ish know a little about the concentration. And then you can also like determine the purity of your end product. If you know like the absolute concentration of protein and you see the on your SDS gel, you see the strength of the bands, you can somehow like plus minus say how pure it is. Otherwise, you don't need to do an HPLC, which is expensive. And yeah, so and then you regenerate the column. So the NaOH is to participate the proteins. The EDTA is for stripping off the nickel. And of course, you need to really wash it good with water. And there are also some other reagents. I will look them up. I will post this protocol soon for column regeneration because they use other steps as well. and really get them super clean afterwards. And then you store it again at the fridge. And the amicons, they're just like columns. You put your fraction in it and you spin it down. Then you put your storage buffer in it, you spin it down, and it takes like 30 to 60 minutes actually, so you can exchange it. And yeah, you also should consider that in your storage buffer, here you have 50% of glycerol, which is not optional this time, because otherwise you get like ice crystals in your storage, and these could hurt the enzyme. The DTT is up to optimization, but 5 millimolar is also the concentration in the NAB buffer, as I read somewhere in a remark, but I'm not sure about that. And with the storage buffer, I handle it that way that I do a twofold stock, and then I use it for buffer exchange with the amicon, and then I put up to 5 the same volume of glycerol in it, and then I can freeze it. And before that, I obviously do a Bradford concentration measurement again. So the amicons, I just show you a quick picture if you haven't done it yet. It's really super easy. So you shouldn't be too, it's not complicated at all.
Are they expensive?
Yeah. So dialysis is more
super cheap or easy to do.
Yeah, it's super cheap. So because I'm not optimizing the purification procedure, so I'm just optimizing the enzymes. So I go with the stuff we have, and which is like the easiest. And here you have a picture. And you can see the filter up there on the left side. This is where you put your stuff.
Still on the protocol. We can only see the protocol screen.
No, I put the link in the chat, sorry. Of a Google picture search.
Okay, yeah.
Yeah, and or I can just like picture search it on the other tab.
And that concentrates your thing.
Yeah, so you put your sample. Just a sec. You put your sample up here. You see, you have here. You screw it out. You put it. You open it, and then you put your fractions in here. You spin it down with a centrifuge, and then it collects in here. And you can spin it down like to a really small amount. You should be over 2 milligrams per milliliter. Otherwise, your protein could participate. But it's also written down at the supplies manual. And yeah, then you do the buffer exchange a few times, and then you diluted your imidazole down so it works well. And then you just put the same volume of glycerol in it, and you're finished after measuring your concentration. And then you should do all the post-processing, the testing, the enzymatic specificity assay, and everything. So, yeah.
I have 2 more questions.
Of course.
The first one is not a question, just I'm getting now the enzyme without glycerol and storing it in -80, because then you can't freeze dry an enzyme with glycerol. So
yeah, that's true. You can also do it without glycerol, and then you just do like the
stored in -80. I think that should still be stable.
Yeah, most probably if you freeze it in liquid nitrogen quickly.
Yeah. Or yeah, okay. I just put it in the -80, and that seems fine.
Okay. Yeah, so probably it's also stable. Because of that, but the easiest way to
keep it,
be nice to your proteins is to.
Yeah, with 50% glycerol, you can keep it in the -20.
Yeah. So
And
the
then-
ice crystals are not cutting too hard.
And I have another question about, like, is there places you can stop in the middle of this, like, insane protocol?
You mean like some breaking points?
Like,
Yeah, there
go
are
home
some.
because, like, the places is closed and, like, nobody. Like, I can't have long days in the lab anymore. So
Yeah,
where
of course.
in what steps can you stop? Like, can you
So-
stop after you lyse and wash the first time?
So you can always, like, when you have the cells collected by centrifugation, you can always resuspend them in PBS, for example, and just freeze them quickly and store them. So I sometimes just freeze them after collection in liquid nitrogen. So you need to freeze them quickly if you want to, like, be nice to your enzymes or to your proteins. And then I just store them at the -20. But I think it should still also work if you just freeze them at the -20, but then you will for sure have a loss of activity.
What do you think if I put them straight in the -80 at that point?
I don't know. I mean, you will always have some activity, I guess. I would say. I'm not sure.
Or maybe in the fridge, like, what if I leave it at 4 degrees? What if I don't freeze?
At 4 degrees, you still have some activity with inside the bacteria, so I wouldn't recommend that.
Okay.
You should always work at 4 degrees with a centrifuge, and optimally you do it in a cooling room. But when you do the IMAC, like the purification at room temperature, it's also okay with this with the BST. I mean, the BST is stable in room temperature for some time, so.
No, I think the 4 degrees, most of it is about the proteases, actually, to get them to work slowly. That's my feeling, but I'm not sure.
Yeah, with the centrifugation, but in the IMAC purification, it's I think at least it's mostly about the
Binding.
About the enzyme itself, but it could be also that it's the proteases. I never thought of that.
Protease, or the temperature will also affect the binding, though. Okay.
Yeah, most probably. I mean, when the binding should be tighter at lower temperature.
At room temperature, usually, like the nickel columns and stuff.
Yeah, I also did it at room temperature. It worked fine, but yeah, it's if you have the option, do it at a lower temperature. If not, then it will also work. Yeah, but it's better with the spinning part. It's better to do it at 4 degrees if you have a centrifuge that can cool down. It would be awesome. If not, you can still try it, but I really would recommend to cool it. Yeah. So breaking points. Yeah. So with the plate overnight, you can always put it in the 4-degree fridge and pick from your plate whenever you want to. So you don't need to do this exactly the same day. When you set up your overnight culture, you have to reuse it the next day. So this is a must. And you have to express them. So these two, so this evening and this day and this morning, midday,
You can stop here in the middle of before
together.
No, we said we can stop after we pellet the cells.
Yeah, so this is a breaking point, yeah, here. After lysis, I wouldn't recommend that because the lysozyme is in there and everything. So I would say this is a safe break point when you just resuspend them some milliliters of PBS and keep them really cool for some time. And after this, so the harvest, lysis, and the IMAC, it's also, yeah, I would count 1 day if you are not like routined in your lab. I would safely count this for a full day of work. You can do the SDS page gel later or in between somewhen. You just need to keep the samples cool, like also cool them, keep them in the fridge at least -20 or -20 works fine.
You don't put 4.
Okay.
You freeze them. You don't put them at 4 degrees.
No, the samples for the SDS gel, I freeze them. And somewhen I cook them with, I mean, they don't have to be like active or something. I cook them in lemon buffer and they are dead afterwards. And after the electrophoresis, they're not surviving that. I never take them out. So this is just for structural detection. Not structural, but for detection and monitoring of the process. And yeah, so you don't need to be too cautious with the SDS page samples. You don't do anything then just looking into it. And yeah, the Amicon, it takes some time with the Amicon. You can do all the steps in 1 day, but you can also, most enzymes also survive 1 week in imidazole elution buffer but it's always depending. So with the BST polymerase, I think it should work if it stays like over 1 day or a few days in the imidazole buffer. But it's always better to process them further as fast as possible, obviously. Yeah, but this is also something.
You freeze it with the imidazole buffer.
No, I wouldn't freeze it then. I would store it at +4. With the imidazole buffer, it's no problem because this is my elution fraction, right?
Yeah. That
so my
purified protein already. That should be fine.
Yeah, so I just have this protein and some dirt proteins or something. So it should be fine to store them at +4. It's better not to freeze them without glycerol too often. So this is totally okay. But don't push it too much because you always, when you, there's always an optimal path and there are super millions of other paths. And the more you divert from the optimal path, the less activity you have, basically. So yeah, and if you want to produce it yourself, you really should stick somewhere consistently to one protocol. And if you change some parameters, you should obviously do all the assays again for activity assay and everything. And yeah, sometimes it also makes sense just to check the quality of your fraction. If you have the assay in your lab working, it's not too much of work. So you should always, like, at least an SDS gel should be something you should run with. Western blot is optional, but it's nice to know when you are starting to implement the protocol. And of course, like, some activity measurement is needed afterwards. Yeah, and then you have your protein. Yeah, here. Okay, so I talked so much now. I don't want to talk anymore. So I'm there for questions, or let's discuss anything. Is somebody there with a question? Ingrid has one.
Okay. I just want to tell Ingrid also, like on this reclone protocols.io, there's a protocol which is similar for expression of a PFU, which is very similar to TAC, which supposedly they got working already in Chile and Argentina too. And they also do a step before where they heat it for half an hour to 70 degrees to take out most of the E. coli proteins.
Yeah, true. That's also like heat participation is something I also heard about. How do you write the protocol? Which protocol do you mean? The one Guy talked about? Okay. It's heat. Guy will provide you the link. Yeah, probably Ingrid, if you want to. Yeah, there it is. Yeah. So another thing is,
and this is a protein that the free genes, like or the reclone people, will send you the plasmid, and also an open pack one. So.
Mm-hmm. Yeah, and there are also other groups in Latin America who are already purifying and producing, at least in small amounts, as I know. And if you, for example, come to the Reclone Symposium, there are always these breakout sessions. And there you can also connect yourself to the others, so you can have some exchange. You can also say something, Ingrid. You don't need to write all the time.
Oh, no, it's just that I have my little girl near me, so he's making a lot of noise. So that's why I'm
And I wanted to, so I'll say one more thing, that I asked them questions on the protocol and they answered quickly. So like you can even write your questions right on this protocols.io. You can ask questions to the people who made it and they can answer.
Okay.
You mean on protocol.io?
Yeah.
Okay.
Is the symposium will be in person? I mean, I don't know what this program.
Nothing in the world is in person now.
No, it's in a Zoom call. So you register yourself on the Reclone page, and then you get a email notification with the Zoom room ID, and you just join by Zoom. So this is easy.
Okay.
Yeah, and always connect yourself to each other. That's why we are all here, basically. And yeah, so if you need some help, you also have our email addresses in the shared notes I will put in the Reclone forum, and you can always write us, and we can pinpoint you to somebody, or just feel free to really ask like basic questions as well. Everybody's happy to help you.